We're going to take a look at the rest of the neurotransmitters. The first group of neurotransmitters is the amines, and remember the amines are made from amino acids, so they're going to be hydrophilic. We have the catecholamines, and remember the catecholamines include dopamine, and then we also have norepinephrine, and epinephrine. And we talked about these before, so these can also serve as hormones in addition to being neurotransmitters. And then the other amine neurotransmitters we have are serotonin and histamine. And these are also like acetylcholine, and they're going to be made down in the axon terminal and stored there in vesicles until the action potential comes down, and then they're going to be released by exocytosis into the synaptic cleft. And then for getting rid of the amine neurotransmitters, we do have two enzymes that will destroy them. So we have monoamine oxidase, which we just abbreviate MAO. And I'm just going to ask you the abbreviation. I'm not going to make you spell out the whole name. And then the other one is catechol O methyltransferase, which we'll just abbreviate COMT. So you don't need to know that whole name. And MAO will chew up any of the amine neurotransmitters. And then COMT is a little bit more specific. And you can see by its name that it's just going to chew up the catecholamine neurotransmitters, so the dopamine, norepinephrine, and epinephrine. Here's the enzyme MAO. Where it's located is in the axon terminal of the presynaptic neuron that released the amine neurotransmitter. And it's actually located on the outside of the mitochondria. And here you can see the neurotransmitter dopamine being released by exocytosis into the synaptic cleft. And then it's going to be pumped back up into the axon terminal, so we'll reuptake it. And then it'll be destroyed by the MAO enzyme in the axon terminal. And then in this picture, we've got the neurotransmitter norepinephrine, another catecholamine, and here you can see it's being released by exocytosis into the synaptic cleft. And here we've got both enzymes that will get rid of the norepinephrine in this picture. So we've got COMT, that will chew up the norepinephrine, and that's located in the synaptic cleft. And then we'll reuptake the norepinephrine back into the axon terminal. And then the enzyme MAO will destroy the norepinephrine there. Okay, so let's just take a look at two of the amine neurotransmitters. We'll just look at epinephrine and norepinephrine, two of our catecholamines. And I just want to look at the receptors that they bind to. So the other names for epinephrine and norepinephrine are adrenaline and noradrenaline. And so epinephrine and norepinephrine are going to bind to adrenergic receptors. So you can see where that name came from. There are two different types of adrenergic receptors. We have alpha and beta adrenergic receptors, and then there's different subtypes of each one. So there's actually 5 different adrenergic receptors, and there's different affinities for how well each of these receptors bind to norepinephrine and epinephrine. Some bind to norepinephrine better, some bind to epinephrine better. You do not have to memorize that. And I'm not going to make you memorize where each of these are located in the body. you'll have to memorize that later in your healthcare classes. So it's important to remember that the adrenergic receptors that epinephrine and norepinephrine are binding to are all G protein linked receptors or metabotropic receptors. So these G protein linked receptors, when the norepinephrine or the epinephrine binds to it, that will activate the G protein, and then part of the G protein will take off, and then usually that G protein is going to turn an enzyme on and will make a second messenger. And then that second messenger usually will activate a protein kinase and phosphorylate proteins, and then that will produce the response. And so in this picture, the phosphorylated proteins are going to be opening up an ion channel. And remember that the G protein linked receptors give you a little bit slower response because it takes some time to activate the G protein and then make your second messengers and phosphorylate proteins and then finally open up this ion channel. So it's a little bit slower response with these adrenergic receptors. Okay, and then just to mention the other amines, we have serotonin and histamine. Both of these are just used in the central nervous system. Serotonin has several different functions, but just to mention a few, it does help keep you awake, and it also is involved in depression. So lower levels of serotonin is associated with depression. And then histamine has a number of different functions as well in the central nervous system, but one of them is that histamine does keep you awake. And one of the ways you can kind of remember that is that if you've ever taken an antihistamine like Benadryl, one of the side effects of Benadryl is that it actually will make you sleepy. And the reason that is that Benadryl will cross the blood-brain barrier, get into the brain, and it actually blocks the histamine receptors. And histamine can't have its effect, which is to keep you awake, and then that makes you sleepy. Okay, so here's just an example of one of the drugs that we use to treat depression. So this is called a selective serotonin reuptake inhibitor, or SSRI. I'm not going to ask you about these, but just you might have heard of these drugs. One of the brand names is Zoloft, which is pictured here, and Prozac is another SSRI. And the way that these SSRI drugs work is they will block the reuptake of the neurotransmitter serotonin. So when serotonin is released into the synaptic cleft, normally it's going to be reuptaken into the axon terminal and then will be destroyed by MAO on the mitochondrial surface here, and that's how it's gotten rid of. But an SSRI drug will block this transport protein and block the reuptake of serotonin. So serotonin will stay around longer in the synaptic cleft, and that will increase the levels of serotonin, because with depression it's been associated with the serotonin levels being too low as a cause of depression. And so that'll increase the levels of serotonin, allow it to bind more to the serotonin receptors, and that will help alleviate the depression in the person. So that's how the SSRI antidepressants work. Okay, our next group of neurotransmitters, these are made out of amino acids. So these are hydrophilic because they're amino acids, and there are only 4 amino acid neurotransmitters. And I just think it's kind of interesting that there are some amino acids that can act as neurotransmitters. So they're not just used to make proteins, but some of them can actually be used as neurotransmitters. So these are only used in the central nervous system, so brain and spinal cord. And so I do want you to know which two amino acid neurotransmitters are excitatory. So that would be glutamate and aspartate. And then which two amino acid neurotransmitters are inhibitory? So that would be glycine and GABA. So make sure that you know that. Okay, so let's just take a look at the amino acid neurotransmitter glutamate. And this one is excitatory in the central nervous system. And you'll be able to see why here. So glutamate, being an amino acid neurotransmitter, being hydrophilic, will be stored in these vesicles, and when the action potential comes down to the axon terminal, that'll stimulate its release by exocytosis into the synaptic cleft here, and glutamate will then bind to its receptor on the next neuron here. And there are two glutamate receptors. One of them here is called an AMPA receptor, which you don't need to know, but you can see that this AMPA receptor is a ligand-gated ion channel that's specific for sodium ions. And so sodium ions coming into this next neuron, what is that going to do to the membrane potential? So hopefully you said it's going to depolarize the membrane potential, and we're going to create an EPSP. So a depolarizing graded potential in this next neuron. And if we let enough sodium in, then we can reach threshold and then create an action potential in this next neuron and then send the signal on. There is another glutamate receptor as well. This is called the NMDA receptor. This one has a little bit different effect, and this one is involved in chronic pain. So when you get to your healthcare classes, you'll probably be talking about this NMDA receptor. And so you'll learn more about that one later. Okay, and then let's take a look at the GABA receptor. So remember, the GABA neurotransmitter is inhibitory, and let's see why that is. So GABA will bind to a ligand-gated ion channel that is specific for chloride ions. So when GABA binds, we'll open the gate and we'll let chloride ions into the neuron. So what is that going to do to the membrane potential of this neuron here? So hopefully you said that it's going to hyperpolarize the neuron. So we're going to create an IPSP, so a hyperpolarizing graded potential in this neuron, and that would be inhibitory. So we're not going to get close to threshold, and we're not going to trigger an action potential in this next neuron, and so we won't send the signal on. So that's why GABA is an inhibitory amino acid neurotransmitter, because we end up letting chloride ions in. And what's interesting is there's a group of drugs called the benzodiazepines, which you might have heard of. An example of a benzodiazepine is Valium. And these drugs will bind to the GABA receptors. So if you give a patient Valium, it will bind to the GABA receptors and let chloride ions into those neurons in the brain and inhibit those neurons. And so you would give a patient Valium, for example, to decrease their anxiety. Maybe they're, really afraid of, getting an MRI because they're claustrophobic. So you could give them Valium before that procedure. And this would inhibit those neurons in the brain and calm them down and decrease their anxiety. So, Okay, and then another really interesting thing are neuropeptides. So these are some neurotransmitters that are proteins, and they're going to be co-localized with other neurotransmitters in the same axon terminal. And they're going to modulate the response of the postsynaptic neuron to the other neurotransmitter. And they do that by binding to these G protein linked receptors or metabotropic receptors. So let's just take a look here at what we're talking about. So let's say that we've got a neuron in the brain, and let's say it's going to be releasing the neurotransmitter acetylcholine. And we've got a neuropeptide, oxytocin. And what's interesting is that some of these neuropeptides have some other functions, other places in the body. So they might serve as hormones. And here in the brain, we've also got oxytocin serving as a neuropeptide in some of these neurons. And this helps explain why oxytocin also has the effect of causing bonding to the baby or to your sexual partner, because it does cause some changes in the neurons in the brain that affects behavior. So, and this is why. So when the action potential comes down to the axon terminal and triggers the release of the acetylcholine by exocytosis, we're also going to trigger the release of this neuropeptide oxytocin at the same time. So we end up releasing the acetylcholine, and then we're also going to be releasing our oxytocin as well. And these neuropeptides will bind to these G protein linked receptors. We're going to activate the G protein, and the G protein is going to take off. And most likely it'll go and turn an enzyme on and probably will activate and make some second messengers. And then those will phosphorylate some proteins. And then maybe those phosphorylated proteins end up turning some genes on and off. And maybe we end up, making more receptors or less receptors. And then that will change how this postsynaptic neuron now reacts to acetylcholine the next time a signal comes through, because you've changed the number of receptors here. So that could affect the bonding behavior. So that's really interesting how these neuropeptides work. Okay, and then these neuropeptides, instead of being made in the axon terminal, like a lot of our other neurotransmitters are, these are actually going to be made up here in the neuron cell body. And that's because that's where the nucleus is. And because these neuropeptides are made out of proteins, remember that the genes and the DNA, they code for the proteins. And so that's why these neuropeptides are made up here in the neuron cell body, because that's where the genes are, the DNA. And so then the neuropeptides, once they get made up here in the neuron cell body, then they get transported down the axon, and then they just get stored in the axon terminal until they get released. Okay, and then you don't need to memorize any of these, but they're just interesting to look at. But these are just some examples of neuropeptides. And you can see the oxytocin we talked about, and then you can see some of these others are hormones as well, but they're also being used as neuropeptides too, so they kind of have some, double duty here. Okay, and then you don't need to memorize these either, but just kind of interesting. Some other neurotransmitters, ATP, can also serve as a neurotransmitter. So not only is it a source of energy, and it's also an RNA nucleotide, but it can also be a neurotransmitter. It can be a neurotransmitter in the central nervous system and also in the peripheral nervous system, and it's also a really big neurotransmitter in the GI tract, so it's used quite a bit there. And then there is one neurotransmitter, nitric oxide, that's actually a gas. So that's pretty interesting too. Okay, and then the last thing is just to kind of summarize some of the important neurotransmitters that we're going to be coming back to again later on in some other chapters. So I would definitely know these for the test. So we've got acetylcholine we talked about. And remember, acetylcholine has two different cholinergic receptors that it can bind to, either the nicotinic cholinergic receptor or muscarinic cholinergic receptor. And then make sure you know what type of receptor they are. Okay, so nicotinic cholinergic receptor is a ligand-gated ion channel or ionotropic receptor. And the muscarinic cholinergic receptor is a G protein-linked receptor or metabotropic receptor. And then also know what enzyme breaks down acetylcholine. So that's acetylcholinesterase. Okay, and then the other two neurotransmitters that we'll be talking about again later on, norepinephrine and epinephrine. Okay, and both of these are going to be binding to the adrenergic receptors, of which there are two types, alpha and beta adrenergic receptors. And then we have the different subtypes. There's 5 different subtypes. And then all of the adrenergic receptors are G protein-linked receptors, so they're metabotropic receptors. And then the enzymes that will break down the norepinephrine and epinephrine neurotransmitters are MAO and COMT. So I would know those as well. So this is a nice little study cheat sheet. So make sure you know all of these things for the exam.